morning, dear students. So, we will now move on to the 11th module, where we will be talking about now in place of the metal ions. Now, we will just simply talk about the non-metallic part, which are very important to our life, to our biological system. So, today in lecture number 51, we will be talking about 3 elemental things and its compositions like the carbon, hydrogen and oxygen. These are very simple things what we are studying from or again from our school days, but now we will put all together in some very complicated area where the nature is doing for us is the biosynthesis of non-metallic biomolecules. What are those? Because you do not have the metal ions now. You can have the amino acid, you can have the protein, you can have the lipids, you can have the carbohydrates also. Then we will talk about the formation of small molecules, how you can take, because we all know the example of formation of the small molecules through the consumption of the dioxygen molecule is your carbon dioxide. When I am talking, when I am breathing also, I am producing lots of carbon dioxide into the air. Then how we can get those carbon dioxide for generation of some beautiful anions like bicarbonate and the carbonate anions that we have also studied earlier. Then what are the roles of the different ion channels and the anion receptors. So, these are not basically the concepts, consider them as the keywords. You read it more about what is the ion channel, how the ion channel will be related to the non-metallic biomolecules, what are the anion receptors, something like that. Okay. So, if we have some inorganic element like carbon, hydrogen or oxygen, so the organic chemist can debate with me, no, carbon, hydrogen, oxygen, they are for their thing, that means they are making the organic molecules. But if you have everything, but we do not put the metal ion, that is the environment what we are talking about, and that has very useful role in the biology. So, if we consider all of them, not that the organic chemists who are interested to have the carbon, hydrogen, oxygen, nitrogen, but sometimes sulfur. But occasionally we will bring phosphorus, we will bring chlorine, we will bring iodine also, the iodide ions. What we know that our common salt, the table salt, we take iodine for the formation of good amount of thyroxine. So, the conventional biochemistry, how this particular part or the way we are looking at it in terms of your biological inorganic chemistry, how inorganic chemist can see from their point of view what is the biochemistry from their eyes or their experiments. So, the remaining non-metallic elements like we can have also boron, we can have silicon, arsenic, selenium, fluorine, bromine, iodine, all these, but it is very difficult to consider all of them together one after another in this particular module. So, I will try to focus, I will try to go for attending those parts where you can apply the basic concepts, how to get a small molecule, how to get the corresponding anions and how those anions are useful for us. Because historically when people identified why iodine is so important to us, why the phosphorus, we know much about this, were the first obtained biogenic materials, because the iodine based materials and the phosphorus based materials like the apatite and all these for our tooth and the bone formation. So, using just hydrogen and carbon, that means the organic molecules what we can have, that is hydrocarbons we call. The organic people when they talk about methane, ethane, propane, butane and all these gases also they call it as the hydrocarbon. So, if you get the hydrogen and carbon in hand and if you try to construct or build many complicated molecules, and these molecules are essential for life processes, any life processes, the bacterial life process, the viral life process, also the human life process and the mammalian life processes. So, how to get the protein, nucleic acid, carbohydrates, lipids? There also we need to know about the incorporation, not only the hydrogen and carbon, but also oxygen, nitrogen, phosphorus, sulfur, all these things. So, the very basic things, the way we know the photosynthesis is giving us the corresponding carbohydrates. So, we are already incorporating oxygen over there. It is not hydrocarbon, it is carbohydrate. So, you have extra oxygen over there, but it is a very simple process. In a straight cut process, we are producing those glucose molecules for us. But when you go for many complicated systems, the protein combined with glucose and all these things, and the incorporation of these things in the amino acid residues. So, altogether how many elements we can have? Altogether we have basically 6 essential elements which can be considered as the building block of all biological macromolecules now, not the small molecule like carbon monoxide, carbon dioxide or even the water molecule. Then some balance should always be there. So, biology governed balance and the geological thing and the chemical thing also. So, the earth's biogeochemical cycles basically where from we get the water, because we are dependent on the corresponding geochemical cycle or the geological cycle and something related to the biogenic thing which we call as the biogeochemical cycle. So, they basically move through from one part or one compartment to the other, we call it as a biosphere, or we can consider as a biotic compartment, or some is abiotic compartment, that is biological part is not there. So, what is your lithosphere, the atmosphere and the hydrosphere, but the biological part is definitely there, but they are not controlling much about this particular movement of these cycles. So, we know that is why we can have the carbon cycle from our school days. Again we are learning this. We learned about the nitrogen cycle, but in the next class we will be talking about nitrogen. So, in this class today we will be talking about these three only, the carbon, the hydrogen as well as the oxygen. So, when you have the carbon cycle, how the carbon cycle is there, we are not talking about from the elemental carbon, but what we get which is readily accessible. So, readily accessible thing what we can consider that every time when I am talking in this particular class also I am giving you in the environment the carbon dioxide. So, how quickly you can produce carbon dioxide? So, it is a very simple thing, but if I ask you give me the detailed mechanism, give me the detailed catalytic processes, how you are burning your glucose molecule or the carbohydrate molecule to produce the carbon dioxide involving other metal ion bearing important biomolecules like hemoglobin and myoglobin. So, life will be complicated in that way. But if you consider the carbon cycle, that means how we can go for the corresponding movement of the carbon in one form to the other and then coming back again to the system, which can be from the elemental carbon also, the charcoal, we know the graphite, all these also. So, you can have the corresponding activities like respiration and the photosynthesis. We know in photosynthesis we are consuming carbon dioxide and producing O2, and the reverse is true for our respiration. So, you should always know about the very simple examples which can contribute to your carbon cycle. So, is basically can be part out by anthropogenic activities when we burn fossil fuel, when we go for other activities, and even we just go for some heat treatment for something, we basically disturbing all these things and we are producing more and more amount of carbon dioxide, which is a well known greenhouse gas. By doing so, we are increasing the terrestrial temperature, we are melting the ice, and all these things are happening together. So, what we can have or about the availability or the amount of iron available and the terrestrial surface in the soil, and above the ground it is stored in the forest, while in the ocean it contains a largest pool of carbon near the earth's surface. So, you have the oceanic pool and you have the forest pool, because the forest all we can have, because if you consider that we are talking about the corresponding lignin, the plant material and all these, they are all having carbon, hydrogen and oxygen. So, about the dissolved inorganic carbon, if some time we ask you what is that dissolved inorganic carbon, is not that the elemental carbon, but is the corresponding form, the corresponding species what we can have based on carbon. So, you can have the carbon monoxide, it can be carbon dioxide, or it can be the different carbonates, that means the inorganic anions. So, it is basically the amount is very high, is approximately 59 times than that in the atmosphere which is available to us. So, if you have the small molecule based on these three elements, the carbon, hydrogen and oxygen, what we can understand? While we are studying about the hydrogenases, we have seen there is a very complicated molecule. You have iron center, you have nickel center, nickel center is surrounded by your sulfur ligation, but your iron center have very small molecules as the ligands. One is your carbon monoxide, the CO, another is your cyanide, that means Cn minus. So, if you do not talk about the nitrogen today, if you talk about the oxygen and the carbon, you get the carbon monoxide molecule. How we get that carbon monoxide molecule? For some degradation also. So, either you can have the small molecule or the radicals. What are those radicals? The inorganic anions we call the acid radicals from our school days. We know how to estimate the corresponding anions, like the chloride anion, that is also there in our body, huge amount of chloride. The way we talk about the imbalance in terms of your sodium and potassium in your body, at the same time we should also talk about the corresponding balance in terms of your anion or the chloride ion. We are not talking about the corresponding anion like nitrate or sulfate or any other thing related to that of your sodium salt or the potassium salt. So, when we take O2, we produce CO2. When we burn some material, when we burn our food material also, we produce CO2. So, if you have the CO2, is a small molecule, definitely is a linear small molecule carbon, and the two ends you have the oxygen. How to make it a carbonic acid, a bicarbonate, or the corresponding free carbonic acid H2CO3. So, metalloenzymes are there, carbonic anhydrase we have studied earlier. Then at enhanced rate, enhances the rate of production of the bicarbonate and the carbonate molecule. That means, the carbonic anhydrase is an enzyme and is a very good catalyst for huge amount of turnover, and the turnover frequency is also very high, such that very quickly you can produce large amount of carbonates and bicarbonates in our body. So, rightly you are getting that good examples of the inorganic anions. Only thing that when we first try to know that how you can detect the presence of bicarbonate and carbonate in your some solution, in your some food drink, the beverages what we take. So, they are all carbonated water. So, how much solubility of that free carbonic acid is there, and whether that is releasing huge amount of carbonate, and that particular thing can also get some relief due to acidity in our stomach, or it gives some other impact on your body. So, these simple inorganic anions, we are not going far away, we are talking about the very simple thing. O2 we have consumed, we have produced CO2, and CO2 due to hydration we are producing the carbonic acid, and after deprotonation that carbonic acid will be giving you the bicarbonate and the carbonate anions. So, one important thing we can consider now, if you allow some metal ions to come into this particular point, that means plenty of anions are available, the carbonates and bicarbonates are available. What we can have the corresponding biomineralization process. So, mineralization process is a typical process. We can go to a geologist to take the help to know or understand this nicely what is mineralization. So, you have the anion which is charged one. So, carbonate and bicarbonate having charges of 1 minus and 2 minus. If you try to neutralize it by putting some cation, say calcium, typical example we will be taking now is your calcium. So, we will be forming calcium bicarbonate and calcium carbonate. You are going for initially the charge neutralization, and once it is charge neutralized, its solubility will also be reduced, and it will try to separate out from the medium. So, during that particular separation process, is not that amorphous powder material is separating as a simple powder like charcoal powder or any other dust. It will go for typical crystallization process, because this growth is very slow and very in a steady manner. So, this particular mineralization process is directly related to your crystallization process, and we get this BM or the biomineralization. Sometimes we also call it as the corresponding biominerals, what we get from there. So, everywhere surrounding us the biomineralization process is taking place, but what is the role of the inorganic anions for this biomineralization process, that we will see. So, around us what are those simple examples? These examples are pretty. In our body also we have teeth, we have bones. In the geological world, geologists are interested about the limestone caves and the rocks also. Then the biologists, other type of biologists who are working on it, they can be very much interested about the corals, some are interested in the ant hills. So, this is a beautiful picture from the natural products through this biomineralization process is nothing but as a inorganic chemist, what I see only the calcium carbonate, but other people can see the beauty of the corals, but we know we are just trying to see through our inner eyes or third eyes that how much calcium chloride is there within these beautiful corals and how these calcium carbonates are forming over there through the participation of the carbonate anion. What we produce in our body, nature is also producing in some other ways, because we are going for the respiration in different forms. Also, not only carbon, not only oxygen, the carbonates and bringing the metal ion species like calcium 2 plus. You can have the silicates. So, that particular form in algae and diatoms, carbonates in other invertebrates, and calcium, phosphates, carbonates and all together in different vertebrates. So, we should look at about the corresponding anions, what anioning species we are talking about, whether we are talking about the carbonates and bicarbonates, or we are talking about the silicates, or we are talking about the phosphates. So, we are moving basically, you see carbon and oxygen was there, if you move you will be getting silicon, if you move further you will get phosphorus. So, these are the basic elemental compositions from there. So, what we see now is that if you have a biologically controlled mineralization process, what we call as BCM, and another we will be considering as biologically induced mineralization. That means, now we are trying to differentiate the typical mineralization process, but we are not forgetting the role of the typical inorganic anions. So, you should know a little bit about the mechanism of mineralization process. As I told you, is typically a crystallization process. So, that crystallization process can be there. So, if you consider that okay, we have this mineralization process for that and we get this particular one for this mineralization. So, what we can have? We can have crystallization. Crystallization with some saturation. What are those saturating elements? We were having O2, from O2 we are getting CO2, and from CO2 we are getting CO3 2 minus as well as HCO3 minus. So, these are the processes, that means we are looking for some saturation, that means super saturated solution. Until and unless you have the super saturated solution, you cannot have the crystallization. We are getting is a typical natural process. So, that is why you are not giving any importance for that. When you do it in the laboratory, we are struggling for the crystallization, that means the growth of the single crystals, we pay huge attention. But what nature is doing for us, how the coral reefs are forming beautifully with the ambient condition, the temperature of the ocean water, the pressure, the water pressure, all these things are important. There is no need to go for any other abnormal temperature what we produce or give in the laboratory, abnormal pressure also. So, the natural process. So, a trained mind, a curious mind always try to look at the beautiful chemistry is going on around you. So, how this particular mineralization process, so what we are bringing is when you bring the biological part is typically the natural process. So, it is nothing but the natural level of studying inorganic chemistry. So, if you have the crystallization and you have the typical anions you get. So, why we define is the when we get what is BCM then? When they are directly synthesized at a specific location. That means, if you have the test tube, we know the test tube, and you put something, some saturated solution of your carbonate or bicarbonate, then you put the calcium 2 plus, what will happen? You have to have your answer with you, because whether your this typical surface of your test tube or a conical flask or a beaker can be useful to allow you to grow the crystals. So, when they are synthesized at some specific locations or on the cell, that is why the biology is coming into the picture. The biological cell is there, and under certain conditions we get the process of biologically controlled mineralization. So, it gives basically the corresponding cell. We call it the cellular chemistry, the chemistry which is happening within the living cell. Then the second type, how it is different? The minerals are formed extracellularly, it is forming outside the cell or outside some other cavity as a result of metabolic activity of the organism. So, when we consider that we are talking about the metabolic activity, we can talk about our burning of the glucose. Glucose molecule, when we are burning, we are producing CO2. So, when we have the metabolic activity, we control, we study the metabolic activity, but at the same time we should also look at the corresponding activity of the carbonic anhydrase, and that carbonic anhydrase can take care of the production of carbonate based the corresponding anions like the bicarbonate and the carbonates, and the rate of that formation is typically very high. So, there is no dearth of availability of these ions. So, how we can move now? So, not going for the very big thing what we produce in our body, you go to some microbes only. So, if microbes are available, so biologists, the biotechnologists, they will come and try to tell you something where we can have the corresponding microbially induced CaCO3 precipitation, which is abbreviated as MICCP, or Mississippi you can consider in a very good name, Mississippi, MICCP. So, you talk about the corresponding precipitation of calcium carbonate. So, but our anion, the inorganic part, in this class we are not talking about the role of the metal ion, we are talking about the role of the anionic part, the inorganic part, that means the carbonate part. So, it is very straightforward chemical process, mainly by 4 important steps: amount of calcium concentration, so availability of the calcium, what we are just now we have said that you can add externally calcium solution from outside to a solution of bicarbonate or carbonate. So, the accumulation of calcium is important, not that you can have the corresponding formation of the carbon dioxide. So, the concentration of dissolved inorganic carbon, try to follow the language, is basically the corresponding formation of carbon dioxide followed by its dissolved form in aqueous medium is your bicarbonate, carbonate as well as carbonic acid, because the free carbonic acid H2CO3 can have some solubility in the aqueous medium. pH is also important, because whether you are getting in the acidic form or in the basic form, and the availability of the nucleation sites. Sometimes we know that the crystallization is triggered or enhanced if you have some nucleus over there. So, external seed we consider that you put the seed, and around the seed or over the seed you go the bigger amount of those crystals. Then not only the hydration of carbon dioxide, but also we can think about the urea hydrolysis. Already we have studied only few days back about the urease molecules, the nickel bearing metalloenzyme that is responsible for the production of the corresponding the hydrolyzed form, that means carbon dioxide and ammonia. So, this ammonia is also producing not only your free H2CO3. So, when we are talking just now about the formation of CO2 then H2CO3, now if you get from some other source the corresponding urea hydrolysis, so bacteria is there or urease is there. So, urease is responsible for your hydrolysis, giving ultimately what? Giving ultimately not only the free H2CO3 what we are getting through hydration of carbon dioxide, but also ammonia. So, ammonia will be controlling what? Ammonia will be controlling your pH, which is important. So, now we get, instead of a very free state, you will having now a buffered condition. So, that buffer is important, and that buffer will control many things. So, products basically equilibrate in water to form bicarbonate, because you are producing H2CO3, and 1 mole of ammonium as well as hydroxide ion will be forming, which give rise to the pH increase or the change in the pH. So, there is some error. So, what has been taken from the book or journal, the 4 will be here, it will be NH4 plus. So, we know the corresponding degradation of the carbonic acid, but also when ammonia is available, hydration of ammonia will be also producing NH4 ion, ammonium ion as well as the hydroxide ion. So, that is producing the corresponding available base. So, the biochemical reaction, what is now happening? Now, you think of another anion which is chloride. We are talking about, if we are biased, if you are focused only to the formation of the carbonate based thing, the calcium carbonate based thing, the medium will only precipitate calcium carbonate, but what is happening on the cell surface? So, if you have the calcium, bring the cell, the biological cell, calcium can be tagged over there or attached over there, and that is giving rise to the calcium bound to the cell. Now, you go simple reaction with chloride ion, bring chloride ion, ammonia is already there and carbonate is there, forming ammonium chloride. You are having, we are proceeding towards a ammonium carbonate, ammonium hydroxide buffered medium, and also another equilibrium due to that of your H2CO3. So, the carbonate will come now, will replace those particular thing, is not try to interact with the chloride ions, but will interact with the corresponding carbonate. So, that gives you the cell bound, cell is your template now, the cell bound calcium carbonate. If your coral reef formation is this mechanism, that will be forming over the cell surface or some point. So, when you have the anion in our body, everywhere we talk about the anion receptors. Is when we talk about the cations, the metal ions, we talk about the ligands, but when you have the anions, we talk about the receptors. Receptors are also there which can grab and which can bind many big molecules like transferrin and others. So, in supramolecular chemistry, which are having some weak interactions, the hydrogen bonding interactions, the other non-covalent interactions, and which is forming between the anions. So, what we can have induce all these cellular processes during the development of cancer also, what we see, that is very interesting observation, that anion receptors, which are nothing but the binders of anions, can be studied nicely such that you can have some good anti-cancer therapeutics. So, synthetic chloride transporters basically, so which are moving chloride, binding chloride, which is transporters means transporting, receptors is binder, compensate for insufficient chloride influxes in cystic fibrosis. So, if there is a diseased condition, is cystic fibrosis is related to the chloride imbalance of the chloride transporters. So, how that can be managed by something. So, you have anions, then you have the anion channels through which your anion is passing, and your synthetic chloride transporters are also there, which is binding the anion like chloride or carbonate or bicarbonate and move from one point to the other, is therefore crucial in biological studies. We have already seen in earlier cases also the sodium ion, potassium ion thing. Now, we are talking about the chlorides only, the anion channels. If your ligand is coming, since the ligand gated anion channel, the glutamate, the simple amino acid glutamate, glutamic acid anionic form. So, if it is there, so ligand will be sitting over there, and ligand will opening up the channel and doing something, and your chloride can pass through that channel. So, that is why the ligand gated. So, the person who is there, who is doing your that particular opening up and closing up of the gate is your glutamate. Then some macrocyclic lactone, which are very useful for its activity as the drug molecule, like ivermectin. So, ivermectin is there, huge amount of billions of dollars people make. They are responsible for animal health, our health, agriculture, fish farming, many places, which is very useful also. But if you know that it is related to the corresponding movement of the anions, corresponding movement of the typical inorganic anions, and their corresponding effect on other things. So, in place of glutamate, you can have the histamine. Then histamine is also another ligand. The histamine gated channels is not histidine, histamine. So, remember the molecule what you can have. So, like carbon, hydrogen can also have enormous importance in the biology. Already we have seen the hydrogenases. I request you to study a little bit about all this hydrogen cycle, water involving water, as well as the hydrogenases through that. So, primordial role it has to play for energy transaction, generation of proton gradient in the biological membrane. We know the proton gradient, then the proton transfer also, ATPase also, the proton pump is required for ATPase or ATP hydrolysis in the molecular machine. And the protonation, deprotonation as well as the redox transporters are also dependent on hydrogen cycle or the proton cycle. So, in conclusion, we just very briefly will now tell, you should understand it nicely, that what we are talking about is that how you can get it, the formation of anions and their importance. So, you know the molecules, when you breathe we take oxygen, how it is related to the formation of very important anions, or you are taking something externally like your table salt in your food material, sodium chloride, or from other food stuff, potassium chloride, how chloride will take part in all these cases, or the biology of the chloride ions or the biology of the carbonate ions. Then the connections of all these things, the anions, the anion receptors or anion channels also, then how they are basically giving some helping hand to us to know about the disease conditions. We can talk about then the health, how it will be useful for our good health, and the medicine, whether we can produce some lactone type of thing which can bind very loosely, but is important for transportation for your all this macrocyclic lactone, what I just told you as ml. So, read all this nicely from the Wikipedia page about the anions in biology and the book of Crichton. Okay. So, thank you very much for your kind attention.